Dr. Leslie Weiner.
Our work at USC, recently we've been doing a number of clinical trials. My own work has really focused on the use of stem cells, and I think that quite clearly that will be something in the immediate future. We're very pleased with some of the new technologies which have moved us in a direction away from embryonic stem cells, which I work on, to the stem cells that are derived from each patient. We are very interested in the Hispanic community, since we have about 15% of our patients are of Hispanic origin, and one of our members of our group, Dr. Mespa, has been studying this group as well. So these have been some major things that we're interested in. Like everyone you're going to hear today, we do work together on a number of issues, a number of projects, and I think that my own feeling is none of that would be possible without Nancy's support and your support.
Hauser from UCSF. This is always such an exciting part of the annual Race to Erase MS. And I'd like to just very briefly mention three things. The first is a genetic study that you might have heard about that was published last week. This was really a remarkable study, and it would not have been possible without being funded. And it was funded through the Nancy Davis Center and through friends of the Center Without Walls. Bill Perkins, is Bill in the audience? Bill's outside, and Katie Brass and AJ Brass. Katie is right there. This is the sort of study that would never have found funding through the federal government, because often you have to prove that a study has been done before it will be funded. And here, that the way that a group of genes were used from the mother's inheritance versus the father's inheritance. Remember, we have two copies of each gene, one from our mom and one from our dad. We found differences between the affected, and it could have happened without your support and the support of the Center Without Walls. Point two, you may have heard 2 years ago at the Center Without Walls about a treatment called Rituxan that goes after a cell called the B cell, the basic work for which was developed in large part by work around this table and funded by the Nancy Davis Center. There is now a new drug that is a fully human protein called ocrelizumab that is going to have its results reported in the fall. It is very exciting data. And what's beautiful about this medicine is that it's a medicine that can be given by vein. And the group has decided upon 3 finalist projects that we will all band together with to work on together, each of which we think will change the face of MS research. And also through a new initiative by Nancy, we are now able to support outstanding young people so that when the baton is passed by the older to the younger generation, there will be people ready to take on the mission. None of this could happen without Nancy's amazing dedication and support over the last 17 years.
Hi, I'm Nancy Davis and chairman of the Race to Erase MS. Thank you all so much for your support for being here. It's a very important cause. We had our fabulous event last night that went off without a hitch, which I'm so excited when that happens. We raised a good amount of money, which.
And actually, I think this is my 15th event. I've been around. I was at the ones in Aspen. And it's amazing that I look a lot younger than I did. 17, so 17 years. What am I going to do? But I'm actually originally from Colorado, and Nancy has roots from Colorado, and the Barbara Davis Center is there, etc. So anyway, I also want to echo my thanks and appreciation to Nancy and all that she's done. Let me tell you some of the exciting things we're trying to do at our center. As we know, everybody is different. Even identical twins can be different. One of the things that I think about is that there's like 7 billion people in the world, and there's no two that are the same. When you think about that, it's kind of amazing. And so that means MS is different too in each individual patient. And one of the things that we're working on very hard, and I'll tell you a little bit how we may want to do it with the Center Without Walls, is individual patient profiling, where we have blood tests and might be able to tell an individual person what type of MS you are, where you are in the disease process, and which drug might be good or bad for you. And we're trying to combine new blood tests that we're working on that could just be done on a little bit of serum, MRI imaging, genetics, etc. So that, you know, when you go to the supermarket and there's those barcodes, so we're trying to come up with like everybody's individual MS barcode so we could tell.
So my name's Kaplin. I'm working with Johns Hopkins. MS has the highest rate of depression of any medical, neurological, or surgical disease. Suicide is the 3rd leading cause of death across the entire age span, lifespan with patients who have MS. Suicide is 7.5 times as common in people with MS as the general population, and it is absolutely preventable, because the good news is that depression, while it is potentially lethal and it is the #1 correlate of quality of life in patients with MS, is whether they're depressed or not, it is absolutely treatable, and it is only treatable if you recognize it and get it treated. Unfortunately, all too often, it gets written off as, well, you're depressed, but you'd be depressed too if you had MS. But look at David, and he's got MS, and he doesn't strike me as someone who's depressed. He strikes me as someone who's sort of a singing antidepressant, as is Nancy.
Put that in my bio.
A source of tremendous inspiration to everybody, I think. And you know, I'll get calls from Nancy periodically, as I did last week. She's rescued someone else who's very depressed and didn't realize that this is much like the other aspects of MS, just a product of the inflammation in the brain. Now, I say that to you, and it's only as of last week we actually have evidence now that just last week we were able to show that using a special way of sort of reconfiguring a magnetic resonance imaging machine, an MRI, to, instead of showing us water, which is where you see those beautiful structures in the brain, we can reconfigure the way the data is analyzed, and we can actually look at the chemistry of the brain, and we now have a chemical readout looking at a particular area of the brain, the hippocampus, that actually shows us a very high correlation between depression, if you look at one particular part of the hippocampus, and if you look at both, there's 2 hippocampi, one on each side of the brain, and if you look at the hippocampi on both sides, you show very high correlations with cognition and memory and concentration. So why is that a big deal? Well, it turns out, and I'm a very proud psychiatrist. Psychiatry sometimes is sort of the red-haired stepchild, and I will tell you that much as Matt is demonstrating the power of technology through Google and TiVo and the like, it was actually Hans Berger back in the early 1900s, a psychiatrist, who said, you know, these people who drop to the floor and are seizing, I don't think they're crazy. I think that they're having something going on in the brain. So it was Hans Berger, the chair of psychiatry at the University of Jena, who invented the EEG machine. And the EEGing and measuring it hopefully will have a way of better, not only sort of having people appreciate it and understand it much as the EEG did for epilepsy, but we'll have better ways of treating this now. The other thing I wanted to tell you is that we now also have not only the evidence that MS through its inflammation causes the process of trying to get together a study to show that these are actually neuroprotective and can improve the outcome of people with MS. And last